Good morning. Welcome to the Bureau of Economic Geology's Austin Earth Science Zoomerama. I'm Linda Ruiz McCall, the Bureau's Information Geologist and Resource Center Manager. Please note this presentation is being recorded and will be posted on the Bureau's YouTube channel for on-demand viewing. Before we get started, I'd like to take a moment to thank our sponsors Equinor and Austin Geological Society for their generous support. So welcome to our guests. I see some friends that have joined us before. A shout out to the Eco Explorers and to our new friends in the homeschool group. Thank you for joining us. Today's presentation is from two paleontologists from the Jackson School of Geosciences of the University of Texas at Austin. One is an expert in non-vertebrate paleontology and the other in vertebrate paleontology. Dr. Lisa Boucher is currently the Director and Curator of the Non-vertebrate Paleontology Laboratory in the Jackson School of Geosciences at UT Austin. She's a paleobotanist specializing in Cretaceous macrofloras and has collected fossils in several regions of the US, as well as field research in Antarctica and Madagascar. Prior to her position at UT, Lisa was an associate professor at University of Nebraska-Omaha and a postdoc with the Smithsonian Institution's National Museum of Natural History. Sarah Davis is a Ph.D. in the Jackson School of Geosciences. She studies the evolution of theropod dinosaurs, including birds, and primarily works on fossils from South America. She's also interested in how birds make their bright coloration and studies pigments used in living birds. Fossils are evidence of past life, but how much information do different kinds of fossils reveal? In today's presentation, we'll learn how the rock record preserves a wealth of clues on how extinct organisms interact with their environments. In the talk, what can the fossil record tell us? And now, Lisa, the floor is yours. Thank you for joining us.
Great, thanks. Good morning. Thanks for joining us, and I'm really happy to share and discuss and talk about life essentially from the past. And so as mentioned, I am an expert in essentially non-vertebrate paleontology, but my actual research specialty is specifically in paleobotany, which is the study of fossil plants. And therefore, I have a background in both biology as well as geology. And one of the groups that I study or focus on is the origin of the flowering plants. Many of you may be familiar with flowering plants. Those are the most diverse and abundant group today, and they reproduce using flowers. In order to study these, I like to go back into the past and reconstruct past landscapes using both fossil leaves as well as petrified wood to study basically how plants change through space and time and how that led to our current flora. So since we have to step back into the past, I'm going to give you a little orientation on the world of the past. One of these is being a little bit more familiar with looking at time intervals of the past. So we're going to focus on Cretaceous time interval. That's part of the Mesozoic era. And if you look on the left-hand side, this gives you an idea of the time interval that this period was. So the Cretaceous spans 66 to 145 million years ago. And during this time interval, the world looked very different. If you look on the right-hand side is a reconstruction from the late Cretaceous, and you can see that certainly sea levels were much higher than today. It was also a globally warm time interval. And you can see that much of Texas was underwater. And so during that time, the sea levels rose and fell. So in Texas, we find both sites that have both marine settings as well as terrestrial settings. And today our presentations will really somewhat focus on the terrestrial environment, but in a lesson activity later, we'll also talk about the marine settings as well. So the question I mentioned I was interested in terms of my research is looking at a shift in the time for different types of floras to have evolved in the past. So we went from a world just before the Cretaceous that was dominated by pteridophytes, which are ferns, and gymnosperms, which are things like conifers or pine trees that reproduce by pine cones. And you can see a diorama on the upper left that reconstructs that past area. So you see some ferns down here. Some of the conifers were larger trees. During the Cretaceous, the flowering plants arose and they began to diversify. And you can see on the lower right-hand side the diorama that illustrates where you begin to see flowering plants like this water lily or lotus families floating in water along rivers. You still have ferns as well as conifers also within the environment. I'm really interested in how this shift happened, and it's really dramatic. So here's a figure that shows you fossil leaf floras through time. And the y-axis is the amount of percentage of that particular group, and the x-axis is time. And so we're going from older time on the left to younger time on the right. And you can begin to see that angiosperms take off in abundance of composition. Now, in order to reconstruct the past, we have to be aware of the limits of the fossil record. Not everything gets into the record. And so if you, I'll orient you to this diagram. If you look at the big spheres that are colored in gray, when you think about living organisms, the biosphere is shown in the upper portion of the diagram. That would be the living, kind of like the part of the world that's living. The lithosphere would be what's in the rocks or in the earth. So when you think about something getting into the fossil record, well, essentially the organism needs to be, a part needs to be discarded or dies. Then you have the organic remains, but it doesn't immediately go into the rock record, the lithosphere, right? It goes through all these series of filters. And so we get organic remains that might get buried immediately, or maybe they're exposed and then buried later. And that really impacts what we end up seeing in the record and how we can interpret that past. And so as paleontologists, we have to really be conscious of our sample and the limits of our sample. That being said, lots of amazing material gets buried within the lithosphere, and that allows us a window into the past. A lot of that process of how something becomes a fossil is called taphonomy. And so when we think about looking at this feature, there are lots of biases in the record. So some of that has to do with our own sampling effort as paleontologists. It has to do with the energy of the environment, and that falls in the realm of the earth sciences of sedimentology. So whether or not something gets buried has to do with whether or not a sediment can cover it. And then also it has to do with biological features. When you think about a feature of an organism, what do you think would actually end up getting in the record? And that usually has to do with how prevalent that feature is or how delicate it is. So as you can imagine, something delicate like a flower is not as often, won't as often be preserved in the record, but something that has a hard shell or a bone may end up in the record more often. As a paleontologist, we also need to know something about geologic setting. This helps us both find fossils and also understand their past environments. And so a lot of my material that I study is of plants, which end up being preserved usually near water sites, because that's where a mudstone can cover the fossils themselves. So for example, some of the areas that I study were past floodplain deposits. Some were near shore, like deltaic environments. Others were in near riverine areas close by that ended up being covered in sediment. And when I study material, I look at both silicified wood that often is in place where the trunk is upright in the deposits, and also the leaf remains end up being preserved and pressed in the rock. And the sites I study often have other invertebrate and vertebrate remains. So we can tell a lot about the biota of the areas. Some of the places I study are in New Mexico, in northwestern New Mexico. And I also am beginning a project in western Texas. Some of the data I'll show you today is from New Mexico. So I study an area that's called the Bisti-Denizin Wilderness Area. It's in northwestern New Mexico. And as you can see, this area today is what we would call a Badlands area, where you can see the bedding plains. You can see that there's a lot of area to hike, and there's abundant fossil remains here. So I mentioned that I study leaves, and this is what some of the leaf material looks like. So essentially these leaves have fallen and were covered by sediment. In some cases, you can see that their dark color preserves some of the carbon remains. And you can see some detail about the shape of the leaf as well as the veins of the leaf. And this helps us identify the leaves themselves and give us features to place it in family. It also gives me an idea of how abundant each type of flora with regard to their leaves was. You can see on the lower right-hand side, this leaf also gives us a window as to what types of insects were in the setting. You can see some insect damage on this leaf on the lower right, where you see holes in the leaf. And so we also look at those kind of biotic remains. So how do I study the leaf floras? Well, I try to collect a lot of them for sampling. I try to ID them based on their features, and I use a method that even modern day ecologists use to sample an area, and it's called the line intercept method. So I take my rocks, like you can see on the right-hand side, I take my blocks of leaves and I count them by increments to look at how much of the leaf was in that particular setting. And at the same time, I can, as I ID them, I have quantified their abundance. And so far, what I found in some of my studies is that the flowering plants were pretty abundant, but only at sites that we call disturbed sites. So near channels where the sediment and the setting was constantly disrupted, kind of like how weeds grow today in disrupted environments. We also have ferns in the record and conifers, but they tended to be abundant, usually away from channel sites, away from the rivers. And ferns were more abundant in the areas that are referred to as distal bank swamps. So waterlogged areas, but not in disturbed areas. Again, this gives us a window as to where flowering plants were doing well in the past. And I hope that allows me to understand how they became dominant today. I also mentioned I studied permineralized wood. And in these cases, we can see an in situ trunk here of permineralized wood. And this wood is really well preserved. The reason it's well preserved is that these were buried pretty rapidly, and they had a silica source that infiltrated their cell outlines to be able to allow for a three-dimensional preservation of the piece of wood. So I can see their cell, or the remains of the cells, the altered cell features to identify that wood. And to show you how well preserved these are, you could take a thin section of the rock and look at it in the microscope. Here's Cupress oxylon, which is a conifer wood, something related to pine today. And you can see in the upper left-hand side, you can see those outlines of the cells. These were the original cell walls. There's no cellular material left inside, but those were the cells that mainly conducted water. And the features on those cells and how they look in the different sections helps me identify it. Another type of wood that we see, because there were also flowering plant trees by this time, here is another genus, but this is a flowering plant genus. And the thing that I want to point out here is these cells are much larger. These were the main water conducting cells. They could move water much more quickly through the plant. And again, allows me to identify those trees that were flowering plant versus conifers. The other thing that I can tell about these plants is with these trunks being in place, I can measure how far away each trunk was. And that gives me an idea of how thick the forest was. So on the left-hand side are some images showing you some in situ wood. On the lower right, there's several trunks shown, and I can measure them out. On the right-hand side, it shows a map that I created at one of my sites that shows the position of the different trunk sites, and I can measure their spacing. And so far, what I found out from looking at some of these late Cretaceous sites in this area of the world is that most of the larger tree stumps were actually mostly conifers, not flowering plants. The flowering plants tend to be smaller in stature, those trees were smaller, and less abundant. I also found out by spacing that the mature trees were pretty widely spaced. So these weren't super dense forests. They were actually more what we would call open woodland. So the tree stumps were further away from one another and allowed space for other shrubs and smaller plants. The tree heights, though, were consistent with a subtropical environment. Again, this was a globally warm time interval, so a place like northwestern New Mexico was subtropical. The largest conifer I found, we estimate to be 48 meters in height. So the reconstruction of that woodland forest would look something like below. And the volcano in the past, or rather the background that you can see, is partially the source of the silica that preserved those fossilized trunks. So that's a general summary, and I think that's my last slide. Thanks for your attention.
Thank you so much, Lisa. That was a great talk. And now let's hear from the vertebrate side of the picture. Sarah, thank you.
Okay. Well, hi, everybody. Thanks for being here today. I'm Sarah Davis, and I'm a PhD student at the Jackson School, and I am a vertebrate paleontologist. My background is in biology, but coming to the Jackson School, I entered the geology program and have been able to combine my interest in biology with geology in the form of paleontology. And I study theropod dinosaurs, which are the group of dinosaurs that include the iconic Tyrannosaurus rex, and also our living birds that we see today. And so my work is really focused on fossils from South America. The picture on the right is me at my field site in Chile. But I also look at living birds. I use living birds as a system to study how color is made, to try to figure out what color extinct dinosaurs might have been. And so that leads me to the topic I want to talk about today, which is really, what did dinosaurs look like? And this is something that I'm really passionate about and is a major part of my PhD research. So like Lisa mentioned, there is a lot of variability in the fossil record. Sometimes we get really beautifully preserved fossils and sometimes not so much. But in low energy environments, animals do tend to preserve better. So that's going to be something like a lake where there's not a lot of wave action going on. And really, sometimes these conditions are so good that the animal doesn't really decompose before it gets buried. So in the case of this four-feathered dinosaur here, he died and he fell into a lake and was covered pretty quickly before other animals could get to it. They couldn't really eat him before he was preserved. And so in these really extreme cases and perfect ideal conditions, we can get excellent preservation, and even things like skin and muscle can be preserved in these fossils. So this is an example of what some of those extremely well-preserved fossils look like. This is from a fossil locality in China. And in this case, we have a wonderful range of fossils. We get things preserved that really don't typically fossilize all in one location. And that includes things like bugs. Here's a bug up here. We've got soft tissue preserved on this fish, and on this guy, he's got some fuzz here. And even on our little dinosaurs, there's some skin tissue around the bones. Even in the fish, we can see some scale detail. And in this case, this is an extinct bird. And inside right here, this is the preserved contents of what it ate. So we get this really fascinating and exceptional preservation in lucky cases. And so this is a really good way to have a window into what ancient environments looked like. It gives us an idea of who was in that location, who they might have been interacting with, and we can even start to get a better idea of what these animals, what these organisms looked like when they were alive. So here are some other examples of this amazing fossil preservation. Here on the left, we have a feathered dinosaur. You can even see the outline of its wing shape preserved here. This little guy, Psittacosaurus, he's a relative of things like Triceratops. He's got amazing skin preservation all across the body, even these little fuzzy bristles on the tail. And if we were to zoom in on this fossil, we can start to see things like muscle fibers. And even when we don't get an entire body preserved, we can still get chunks or little pieces of good preservation like we have here. This is a little tiny piece of skin from a hadrosaur dinosaur, a plant-eating dinosaur, and you can see that scaly, bumpy texture of its skin. And so, along with this amazing data from fossils, we can also look at what living animals look like. We can see the structures on living animals and make comparisons with what we see in the fossil record to make these reconstructions. And so, for example, this is a living bird called a cassowary. It has this amazing structure on top of its head. We see a similar structure in other dinosaurs, and we can start to make these reconstructions. Even in this Triceratops, it has horns, and we can start to see evidence of these keratinous sheaths like we see in living deer and antelope. So we can start to, again, make these comparisons and reconstruct what this animal might have looked like. And even T-Rex, who has been the subject of study for many, many years, we're still finding new evidence for what it might have looked like when it was alive. So we can actually look at the skin of crocodiles and alligators and see similar structures that we might expect on T-Rex when it was alive. And part of why I'm really interested in theropod dinosaurs is because we do have living representatives of this group. Birds are living dinosaurs, and so we can study the diversity of birds to get a better idea of what theropod dinosaurs might have looked like. So if we look at our living dinosaurs, we see a really wide range of colors, of habitats that they live in, things they're eating, ways they interact. And my favorite part is just how diverse in coloration they are. We've got really bright colored guys down here. We have more browns, but all of this is fascinating diversity in coloration. And so by studying the color of living birds, we can start to get a better idea of what extinct dinosaurs might have looked like. So for example, we can look at these two incredibly preserved fossils. These are both little theropod dinosaurs. They both have feathers preserved. Here on this guy, he's called Microraptor. He has wings on his arms and his legs even. And this guy is called Kaihong Juji. He has this really fascinating fan-shaped tail. Well, what we can do is look at the feathers of living birds. And when we zoom in on the feather structure of these birds, we see these teeny tiny little pigment cells called melanosomes. And different shapes of different melanosomes correspond to different colors. So this kind of shape corresponds to a black color. These little dots are a brown, and then these little pill-shaped melanosomes make gray colors. So we can study that structure in living birds, and if we have exceptionally preserved fossils like we do here on the screen, we can take little teeny tiny pieces of those feathers, zoom in with a microscope, and see similar structures in those fossils. And that allows us to actually reconstruct what color these dinosaurs might have been. So Microraptor, we see similar structures in the feathers that we see in living grackles that you might find in your backyard. And so we can reconstruct Microraptor as this kind of shiny, iridescent black color. And then with Kaihong, he had two different types of melanosomes in his feathers. This one along the body is more like what we see in crows. And then along the neck and head, it was the same shape melanosomes that we see in living hummingbirds. They give this really shiny rainbow color. So we've got a rainbow colored neck and a black body. And we've been able to do this for a lot of different types of dinosaurs from all different groups. And we get this really rich picture of what colors extinct dinosaurs might have been and what patterns they might have had. So this guy up here probably had a raccoon striped tail, more of a brown color. That Psittacosaurus with the fuzzy tail, he was also a really consistent brown color with these more yellowy bristles. Anchiornis is another feathered dinosaur that was black and also had these white stripes with little red mohawk. And then even these really large dinosaurs, these armored dinosaurs, we're starting to get a better idea of what colors they might have been. And so all of this evidence together really paints a picture of a bright and colorful past. Dinosaurs were not only very active, energetic animals, but they also had a wide diversity in appearances. And using fossils that are really exceptionally preserved is a key into figuring this out.
Well, thank you. Thank you, Sarah. And Lisa, that combination of vertebrate, non-vertebrate was so fantastic. I appreciate it. And to our audience, in just a minute, I'll be unmuting you to see if you have some questions. We'd love to hear them. and I really liked about your talk how it proved one of the main concepts, big ideas in geosciences is that the present is the key to the past, and how you used the combination of learning and being experts really in current life forms and comparing that to past life forms to interpret them. That's amazing in both fields. I will go ahead and start with the Eco Explorers. If you would like to ask a question, you may unmute yourselves.
Hi.
Hi.
I have a question
This is Cora.
about dinosaurs. Where do you guys usually look for the dinosaur bones most?
Oh,
That's
good question.
Really good question. Yeah, it's really going to depend. So I mostly work in South America. My site has been only recently discovered. So when we go out and look for fossils, sometimes we're really lucky, sometimes we're not so lucky. We don't have a great idea of where the fossils actually are in our locality, but we really pay attention to the kinds of rocks that are around. We're going to look for rocks that were mostly formed by things like lakes or streams or rivers, something that's close to land. Most of my fossils actually come from what's called a delta. So that's a really near shore ocean environment. So we get mixes of ocean fossils and the dinosaurs. But that's where I typically look.
That sounds cool.
It's very fun, yeah.
Owen, do you have a question?
Hi.
Hi, Owen.
Do you have a question? I have a question. If we wanted to just kind of pack up and go out and look for some fossils or just kind of dig around, do you guys have any suggestions for maybe where to go or how to get started, just on our own homeschool level?
I'll answer that. At least the Austin area, like really close by, a lot of the time interval exposed here is going to be a marine environment. So the Shoal Creek area, there's a few places I can send you the info to, but there's a few places where an organism called Exogyra and some other Cretaceous marine fossils are exposed on the surface that you can go collecting. So there's some local Austin sites for that.
Cool, thank you.
I wish there were some dinosaur and more plant sites nearby, of course.
Yeah, that would be good.
And yes, there are some wonderful parks that have preserved fossils that you can see. I'll have to say it was really fantastic that there is a national monument in Waco, Texas, that is a mammoth site, and that is a great place to go. And it's a national park here in Texas. I'd recommend that. And there are other places. We have a publications store at the Bureau, and there's some guides for fossils. And what I really took what Sarah said very seriously, in that there's an interplay between paleontologists that are geoscientists, and there's also other disciplines where they study the rock units and what types of environments they were deposited in. And I'm sure when you look for fossils, you draw upon the knowledge from other geoscience disciplines before you go out in the field. You look at the maps and interpretations of environments before you go. So it's a very interdisciplinary science. Let's see. So I have the Eco Explorers on the line.
Hi, how are you doing, Linda?
Hi, great that you guys are joining us. Welcome.
my guys are being shy. They wanted to share that there are theropod tracks in the San Gabriel River, just north of Austin, off of 183. You can actually hike up the river and see a set of tracks there. We wanted to share that with the other family.
Oh, thank you. And you have access in streams that you have to be careful because it's some private property laws. I have seen those tracks. It's quite impressive. They're like 20 by 15 inches, some big three-toed dinosaur tracks that are just right in the creek bed.
I have a link to a blog about them. If you guys are interested, we can also add that to the resources.
Oh, great. We'd love that. Yes, thank you. And, just another, yes, please.
Oh no, we were also going to say that at Walnut Creek is also a great place to, there's a couple ammonites there that are easily accessible.
Yes, you guys have been collecting for quite some time, and that's very exciting. I think that inspired me when I was a child growing up near Austin in the Cretaceous beds. We'd find fossils, and it's just wonderful. So I wanted to ask you, Lisa and Sarah, both. Lisa, if you'd start, what inspired you to go into your area in paleontology? What was your path? Because in Zoomerama, we want to talk about careers that might inspire young people to go into the geosciences.
Yeah, I think that, I didn't start out by thinking, oh, I'm going to be a paleontologist. It was more, when I was a kid, I think that I was interested in biology in general. So I was, where I grew up, I was a little bit more in the country, so I like to be outside, collect things. But when I decided to go to college, I definitely was already interested in biology. But I wanted to do like medical research or that type of thing. And so in college, though, I started working in a lab, and the lab that initially hired me as a freshman was a plant, they were a plant virology lab, which got me curious about plants. And so then I took some elective classes, and then I realized, oh, wow, plants are really interesting, cool. I want to know more about them. And then I got more interested in their history. And so by then I pretty much decided I want to go into science. And then it became a matter of, okay, I think I'm going to study plants. And I wanted to know about how to ID them and their history. So when I applied to graduate school, I looked at labs that both had programs that had modern plants as well as fossil plants. And then I got hooked. And I think the other major thing for me is that I love to travel and be outdoors and hiking. So I love to collect fossils, like the whole collecting aspect, whether it's modern or fossil, involves that. And so that basically drew me in as well. But really, it was just a burning curiosity about things that were unusual.
Ah, that's a good story. And Sarah, how about your interest in paleontology? How did that get started?
So really similar to what Lisa said, I was always really interested in the outdoors and looking at animals, looking at how things worked. And I went to natural history museums all the time when I was a kid. It was my favorite thing to do, to go see the dinosaurs and visit museums. And then as an undergraduate, there really wasn't a paleo program at my school, but I knew I liked biology and I wanted to study animals. So I went into bio. I started working in a biology lab that looked at bird populations and how coloration has evolved in a specific bird species. And so from there, I started looking at graduate programs, figured out that paleontology was a real thing you could do. And so kind of started talking to people and found it was going to be possible to combine this interest in color and biology with the paleo side of things. And then when I joined the department here, got started with field work, kind of got introduced to that side of the field and just fell in love with it. So now have decided that I want to do both field-based work and lab-based work, and have gone from there.
Ah, that's great. So much creativity involved, but you guys directed your own paths. It sounds like you both found, I heard you say, I loved this. I found that I loved that. And there's a philosopher, Joseph Campbell, who said, follow your bliss. I'd recommend that. If you work in something that you enjoy, it makes your whole life a lot more enjoyable because you're doing what you like. And I'm glad you guys shared that. Thank you. I understand you guys have an activity now. We have a website for this topic on our Zoomerama pages, and we have educational resources that you can access, and we'll also have a post of the video. But Lisa and Sarah, would you share a little bit about the exercise we'll be posting on the website, please?
Sure. So we've kind of come up with this activity where you get to be the paleontologist, and we give you some fossils that you found at your field site, a little bit of background information, but then the activity is really based on you interpreting what this fossil environment was like when all the animals were alive. and making connections between different animals and plants and how they might have interacted. So there is a Word document or a handout for the activity, as well as another PowerPoint that you can go through with images and some more information. And it's all based around a real field site in Texas. So it's actually based around fossils that have been found around Austin, as well as in southwestern Texas in Big Bend. So they're actually fossils that you could find potentially around Texas. And if you want,
great. That sounds like fun, kind of a mystery. It's like solving a mystery.
Yeah, kind of some forensics.
Good. So I encourage you guys after this, check the webpage out. We'll probably have it posted later this afternoon, so you can have those exercises to do at home. And I think at this point, we're going to conclude this meeting. It will be, as I said, we will post the video on YouTube and on our website. For those of you watching this on YouTube, if you have a question that you didn't have answered, please leave a comment down below and we'll do our best to get back to you. Also, check the description on the video links for more information to our website, the Zoomerama website, the Bureau website. And don't miss our next talk, Rock Your World, today at 2 p.m. Central Time with John Michaels. And soon that will be on YouTube as well. Thank you all for watching, and we'll see you next time. Thank you. Bye.